Right. Well, while we're waiting, firstly I'd like to say that there have been 6 very entertaining and informative speakers and talks. And from my perspective in the NTD space at least, there have been huge advances in screening cascades, mining of chemical space, and also in collaboration. What I'd like to do now is prompt a debate. I don't want the 6 or 8 people here to be a panel to answer questions, but really to seek your opinions and debate. And I can certainly say, looking around the room, I can see a number of opinionated people, so I'm sure that shouldn't be too much of a problem. I've put up and behind me here just a few points, and I'm sure we won't cover all of these, but they're just to prompt some ideas, and people may want to open that. I'll start with one, and really a couple of points on biology, the zone where I feel a little more safe. And in terms of the models that we're looking at, and I'll start with the models both in vitro and in vivo. There are many reasons for making models, but for us, it's really predicting efficacy in humans that we're after. And particularly with the parasites we're looking at, Leishman cruzi, I think the number of strains and clades that we're looking at probably represents quite a number of species. And we're looking for candidates which hit all of those strains, often using strains which are well adapted to the laboratory. And one question I've got, I'll have 2, one in vitro and one in vivo, is in looking at all these strains, is this resulting in a loss of good candidates? But more importantly, if there's a compound series which is not effective against all of the strains, what would we consider to be a defining assay that would enable us or give us the confidence to go forward to the clinic? Would it be the primary isolates or the host cell or whatever? And a second in animal models, so I'll open this up, I'll just put, so it's biology in 2 points. First is in vitro. What would we consider to be a defining in vitro assay for Leishman cruzi? And for animal models, again, we're looking at strains which have been adapted to grow usually in an unnatural host. And should we use this animal model as what some people call a furry test tube or an animal assay, I think Graham uses, combined with good distribution, metabolism, PK data to go into people? Or should we try to mimic the disease? Or finally, I think in this point Jorge has made at times, should we look at natural infections that are related to the human disease? So I think on that 2 points, I'll leave that open to the room if anyone has comments about what would be a defining model, or anyone on the panel to start that up in terms of the clinic, and how far should we go with animal models before we make a decision to go to the clinic? Started slowly on that one. Yeah, Lucio, please.
I didn't put in that black box the animal model. For example, for cruzi, I began to ask in Brazil, so there are so many people with different animal models, different strains. Really, you can get a very good result, because when you test, you bring your compound, you test this animal model, you have a negative result, you move for the next one, you go until you get something that gives what you expect, And, testing mouse, I don't know how close is this from us. My point is, for at least for Brazil, I'm trying to organize these people together, to put everybody in the same room and discuss this. I want to have opportunity to get good leads and ask these people to try in these different models, but in conditions that you can control, the experiment done in the same place by the same person to compare these models, Because I think this is a really bottleneck,
Right. Perhaps please, yeah. We have a couple there from the back. I think if we keep one mic down here that we can pass up and down, we can probably...
And if you don't mind, say your name before you ask your question, please. That would be helpful.
Hi, my name is Bill Speed. To me, a fundamental question is, what are you using the animal model for? There are many examples where we've cured animals and then we've taken those compounds into the clinic and they haven't worked across a whole range of diseases. So, yes, we can design animal models. We can take those compounds into those models and we can show efficacy. Are we using those models as a surrogate of human? Are we using them to try and define a target concentration that we want to achieve in the clinic? Understanding, I think, the fundamental reason why we want, what we're using the animal model for, almost come backwards and think then what is the appropriate animal model that we should be using to achieve that criteria. So to me, that's the fundamental question to ask. And again, from in vitro, what are we using the in vitro data for? Clearly, an in vitro system, you may have a completely different set of barriers that you need to cross to get the compound to the site of action than you do when you get to the animal model, which then again may be different to what you get when you get into the human situation. So to me, understanding all of those different criteria helps you understand what screens, what models to use at what time, and then having defined your characteristics of your molecule in terms of predicted human pharmacokinetics, you can then decide whether or not that molecule may or may not achieve what you're trying to achieve based on the animal model data you have. So it's a rather convoluted way of saying you really understand what you're trying to use your animal models for.
Yeah.
So I sort of throw that out to the panel to answer that.
Certainly in Rashmi, I think you have a point as well. Just up behind there.
Rob, your comments on animal models reminded me of my advisor's quote, and that was, man is the best experimental model, or best animal model. So, we all struggle with the animal model data coming out, particularly in the neglected disease areas. And in my previous lives, many times when we were wrestling with such problems, we used to take 2 compounds in the clinic with very minimal toxicity data to pick one based on the data coming out in the species that counts. So I'm wondering whether in the neglected diseases, any thoughts have been given to curtail the amount and emphasis on animal data. While I completely agree that we have to have some animal data for ethical reasons to make sure that we don't put crappy compounds in humans. But I'm wondering whether there are any thoughts given to take 2 diverse compounds based on some encouraging in vitro and reasonable animal data to take into humans and make a decision based on human proof of concept. And as somebody made a comment that these are, for most diseases, with the exception of Chagas, the length of treatment is not very long. So to establish a proof of concept is not going to be that difficult also, So I'm just wondering whether should we be focusing to take 2 compounds in the clinic as quickly as possible, or keep on struggling with the animal data?
I think that's a very good point. If I'll give my opinion, I'll leave it too open to others, but mine is very much that we have such little data from humans with what are considered the types of molecules, I think as Alan pointed out, that we're looking at now, to really validate those models. In my mind, if we look at more than one model, we're probably going to get ambiguous data. So they are of value in terms of getting some idea of PK/PD, but I would personally suggest that with those data we go to proof of concept. One of the problems with proof of concept in many of these diseases is that it's short course treatment, but often the clinical endpoint is quite long, and that really does make it a problem in terms of POC and the need for better biomarkers. Yeah, please, Bob.
Hi, Bob Jacobs from Anacor. I'd like to expand on Bill's comment a little bit earlier about animal models. I think one of the key things that we learned in a number of neglected disease projects is that one of the things that I've seen missing and is now being developed in the neglected disease field is the appreciation of the need to incorporate PK and PK/PD early in a project. Too often, I think, historically, we've gone into an animal model, we find either a success or a failure, and we just jump that compound straight forward into a next animal model without really knowing what the driver of efficacy is. Is it a Cmax-driven effect? Is it a time over MIC effect? Is it AUC? What's the exposure? Is the exposure to the right tissues? I think that's something as we develop new chemical series and approach some of these diseases that we just have to keep in the back of our mind. It's hard work to do, but the pharma industry has done this for diseases of the developed world. I think we as a community owe it to our patients to do this for the neglected diseases as well. And I'd like to hear other people's comments on that.
Yeah, Paul. Just, please do.
I completely agree with Bob, because we'd never do an in vitro experiment without knowing how much material we put into a well. So we have to know what compound there is in there, how long it's there for, and so we can start to actually develop that. I think also we have to have a level of pragmatism, because unfortunately some of the models in this space are very long. And if you're trying to do lead optimization and people work hard to develop what they think is the best model, and it's a 6-month model, that's going to kill drug discovery in this area. So there has to be a level of pragmatism in terms of the models that are produced, and they may not be the most predictive models, but they have to be doable to allow us to move forward. But actually, I think now we can start to, but actually, again, from Bob's point, actually we can now start to date bloods from the efficacy studies, so we can actually do the two things in parallel. So I don't think it's a case of either or, and, we can do that. But actually, optimizing on PK stops you going into these long-term models with compounds that are just not going to work, but just move it forward.
Alan, I certainly agree there, and I think we found that. I think the oxaboryl ferhat was an example there where we were using PK and in vitro activity because the model was 6 months, and it was quite successful.
Yeah, I think we've got to be a bit careful with some of these models, which I think people have selected because they look like they mirror the disease in humans. But in actual fact, some of the animal species you're using do not mirror humans. And I think that the classic case here is for leishmaniasis. Most parasitologists would tell you that the relevant model is DNDi infection in hamsters, and that the infected mouse model is not as good in mirroring the human disease. But from a lead optimization point of view, you can get trapped in optimizing activity in a species which metabolically treats your compound quite differently than, say, a mouse will do, which usually is more relevant to the human situation. So I think you've got to be really careful with your models in deciding whether they actually mirror disease progression in humans and whether that species is actually relevant to what will happen in a human.
Pascal.
Thanks. Pascal Messers, Swiss TPH. Well, I guess that might be a case for leishmania, but for the trypanosomatids, I was wondering, of course I agree it's nice to have soon proof of concept in man, but isn't the discussion rather theoretical, because for the HAT patients are really difficult to reach? As you mentioned, for Chagas and HAT, the endpoints, clinical endpoints, they take a long time to reach. So to me, there's simply no way around the animal models. Of course, the way for Chagas would be to have a chronic model to maybe make them more predictive.
Yeah, I think
at least there are models where we have the same parasite. I'm sort of thinking if you have anti-malarial drug discovery, the situation is even more critical, because there is no good, or there's skid mice, but there's no convenient falciparum model.
I think it's a good point. I think it also argues for biomarkers, because some way of getting a faster result in the clinic, as well as using the models. I might change. Oh yes, Charlie.
Maybe just add a point building on Paul White's comment about needing a a feasible cycle for optimization, and if the models are very long, it doesn't support optimization. I think what we've heard today about some pretty exciting in vitro developments before we think about the in vivo models could help us characterize the activity of our compounds. Do they kill fast? Do they kill across a panel of strains? That information we could probably answer in vitro quite quickly rather than finding out in a very difficult in vivo model. Maybe the in vivo model is just for telling us that with the appropriate barriers, the compound can reach the parasite and kill the parasite. But we've already learned a lot in vitro about whether it's going to be successful. And then building a comment earlier, perhaps we should get into the important species, which is man. So, I mean, it's very pragmatic. I'm a chemist. I want some feedback. I want to move things along. It may not be the best model, but we may have to do something like that if we want to move along more quickly. That's my opinion. I don't know what other the panel members think about particularly the value of the in vitro screens in the predictive sense and coupling that with in vivo data.
Tom.
Yeah, so I totally agree, Charlie, and with other people as well. I think the dilemma we have, though, is that in a pharma environment, there's an organization with a large amount of money that is only doing this work and is completely committed to getting outcomes in that space. In this environment where the funders are kind of outside of the organizations that are doing the studies, I think the cost of a negative clinical experiment is pretty high. And so I know just from my own experience in pharma that the worst possible outcome is a clinical trial that fails for reasons that could have been predicted. You know, where you don't actually learn something that moves you forward. All you learn is that you didn't have a good compound, which you might have been able to guess. And so I think you want to be cautious about saying, well, let's push forward to the clinic, you know, to learn something out there that we can then try to drive back to, you know, to an in vitro experiment or an animal model. I think you can do that, but do it when you're really, really confident that you've got a compound that's going to meet a lot of your TPP. You know, at least, you know, based on the PK side, at least. Yeah.
Rashmi.
I think this comment is very relevant, but this, you know, with all well-intentioned people, the reality is to do a small proof of concept studies in a country where these diseases are endogenous is not prohibitive. So you cannot equate the cost equation with the mindset that in my, you know, large pharma life, this is how I have done, and the cost of clinical trial is this. The realities in India and African countries is very different, and it requires an understanding of what it takes. And this is, I've been on both sides of the pond for now, foreseeable future, and I understand that, and this is the reason why I have made that comment. And while equating this, you have to also equate the time it takes for animal model data to be generated and the cost it is going to take. So, Rob, you know what it takes, how much it costs to do a clinical trial in Bihar.
That's a good point.
Sorry.
Oh, we've got more.
I don't know. Sorry. I go Maria Paola Costi, University of Modena, Italy. I'm going back to the previous topic, sorry for a while, because people went on. So, as it was said, to go to phase 1, we need I think we need much better candidate lead. And this we know when we try to develop a better candidate that in a way, for example, that, to my opinion, could stay on much deeper in vitro assays, for example, or assays on animals, dealing with a more closer biological activity profile. I would like to explain better. I would say that usually to test the activity of one compound at a cellular level, we go directly to the target. For example, in some cases, we go a bit around the target and with some off-target biomolecule that interact with our compound. But probably we need to have a much wider activity profile. That means to have a more specificity. And if we have a more specific compound, and we can monitor with this also the development process, probably at the end of the preclinical work, we have much better compound. This is one thing that I think is very important.
Fine. I think, one more, and then I might change. Please go ahead. And I was thinking of changing topic into one that I'm probably less comfortable, which is, well, not less comfortable actually.
Yeah, I fully agree on the gap in clinical data with animal models and in vitro. I think, but the gap in clinical data is, I think, for me, is critical. We have two recent examples of almost failures, if not failure at all. I wonder if we know why, and is anybody doing something to understand what happened? Because I mean, we have here two clear examples and very recent ones. And maybe because they failed, we are never going to build on this experiment. So we have plenty of experiments ensure that the decision of putting this in clinical trials was based on facts and data, but they were wrong. So is anybody doing something to understand why it failed? Because if we are not ready to do that, why are we proposing more experiments?
Mm-hmm. A good point. So I think one more, Isabella, then I want to change across to open innovation. I think there's a few points I'd like to raise.
Hi. I think, in a way, my comment was along the lines of the last comment. I think that the fact that one indeed went and took the compounds, the azole class of compounds, to human proof of concept, actually, and that one was able to generate proof of concept data, but also PK/PD and also a lot of information, is actually something that would makes us go one step further. And I think now it's up to us, no doubt, to understand, and not only to us, but to understand why it failed and the reasons why it failed. So I think it's probably a balance, right, of improving the animal models that we have, improving the in vitro data. I was talking with Lucio moments before this session, where we were discussing the fact that, well, the panel of strains that we have today, it's a panel that is selected, and we do not know, and we do not have information today on Chagas molecular epidemiology to justify the selection of one strain versus another, actually. And so if one, for example, in this panel of strain would have included, for example, those that are known to be resistant to benznidazole, VL10, Colombiana, we would certainly have a very different pattern of curves with benznidazole that we had in comparison with posaconazole. And so I think we are currently, certainly it's a matter of here of selecting what we will do and very pragmatically select those that we will take forward. But we'll probably be needing to evaluate more models systematically, right, in the in vitro ones, and take more chances to fail for a while as we improve proof of concept design. And I think that the trials that we discussed and have just concluded actually will make us design better trials, shorter trials, and will also feed back into the preclinical work that is done. So it's a balance there.
I think it's very well put to take what clinical data we do get to educate ourselves in the models and be willing to take some risk. I think that's a very, very good point. I'll change the topic to collaboration, and particularly some areas that Thierry and Mark had talked about. So the two areas of collaboration I'm particularly interested in. One is collaboration within and with the pharma industry, and the other is open source, open innovation, drug discovery as it's being put forward. But I'll start mainly within the pharma industry and the Declaration of London, which was a group of pharma companies or CEOs who got together to say they were looking at collaborating more effectively and better in the field of neglected tropical diseases. And what I'd like to put forward is we've looked at some very interesting models of particularly bilateral agreements between companies that have been very, very fruitful in terms of exploring each other's chemical space. There's an enthusiasm, I believe at least, that many companies are now looking at the possibility of multi-partner agreements, more than just two companies working together. And the question I have, and I think partly you've answered it, but if you could expand a bit, is by bringing more companies into the field in a similar, perhaps, exercise around one lead series, are the chances of success going to improve as we increase the number of companies that participate, or do you think there's a limit whereby going further than that we're likely to really not achieve much more? Thierry, or?
I don't know about what will be the right limit. So I think about, for example, screening exploration, I think we'll try to increase. So at the moment, I don't know if it will be advised to that 10 different partners can screen our collection. So I don't know, because as I say, there is always a limitation about the targets of interest. We should not pursue the same target with the same similar compounds and working together. There's something we are competing each other. We are still competitors at the end. So we need to maybe to use this open innovation really to find help where we fail with something new. And then I think that's a really critical point. Then if this partner A or partner B can help on specific things, I don't see why we should put a limit. Again, we take this as a representative, not representative, but just an opinion on one of the AstraZeneca partners. I don't know if this the real, I would say, answer for the all my manager, I would say, but this is what I think. So I would say no real limitation until that is clearly mentioned, target we work and remove any conflict of interest and any clash. That's one way to answer.
All right. I think perhaps, Mark, I could expand a little on a point you made on the decision for Pfizer, was that if you have a druggable target, it's likely that one company will make a drug but many will fail. And the question I have is particularly using the types of ways of exploring database, and as you know, I'm not a chemist, but the methodology you have, do you think it would be possible to bring together a number of partners to try to weed out those who will fail, or give a greater chance to find the one that will succeed with the protocols that you're currently designing?
I think the short answer is we'd like to find out. I don't think we know for certain, but I think we'd love to do the experiment. Mm-hmm. And it won't be easy to do the experiment. In terms of, I think there are ways in which, there's such a large body of data within pharma companies on, off targets, let's say like you've just like HERG or something like that, that to be able to combine all of that data to see whether we could ask the question, if we combined it, do we get to a good place where we can make good predictions about what will hit it and what won't, that are better than we currently have, for example. And I think that's an experiment we'd love to do. I mean, I think, if, when I've talked to a couple of people at pharma companies about, their perspectives on sort of enabling this kind of thing, it seems to me that they are willing to proceed on the basis that it doesn't cost too much, ideally. It's not enabling competitors by doing this. So if it's going to, for sake of argument, to put 200,000 compounds through some kind of fairly expensive assay is an expensive business. It's one thing if that's going to enable people to discover drugs for malaria or a leash. It's another thing if it's going to enable somebody who hasn't made that kind of investment to take a step change in their own productivity. So I guess I would love to be able to see that experiment being tried. And I think, you know, there are people that are getting towards that, and I hope we'll be able to do that.
I'll introduce myself after about my fifth comment. I'm Paul White from University of Dundee. This experiment is actually going on at the moment within the TB Drug Accelerator program, which is a collaboration of about 8 pharma companies and I think 7 research institutes, where the pharma companies are putting their compound sets into the research institutes who can do the TB screening. Some of the companies are doing TB screening themselves. And then the compounds coming from that, the companies then triage it, decide what they will release into the TBDA, and a kind of a confidential within that. And then those structures are going to other pharma companies who are looking in their sets to see whether they have related compounds. And then they're releasing those compounds blind to be screened to follow up those hits. And then if those compounds look interesting, then they look to release those structures. Also, what's happening is actually companies are inputting other things like safety pharmacology screens, DMPK screens, medchem input. They're releasing intermediates for people to run chemistry. Companies begin to release intermediates to us to run chemistry in the TB space. And they're running safety pharmacology screens and things, DMPK screens for us in that space. And then the compounds are going into animal models, and GSK are very, very generous in terms of running a lot of acute TB models. So, and then there's other groups are doing going back and trying to find the targets of those compounds within the kind of research institutes. So this is happening. I suppose it's underpinned by Bill Gates beating up some of the company CEOs to kind of contribute funding from people like the Gates Foundation to kind of bring it together. But the problem you find, the more companies put in their compounds into the mix, actually the more resource you need to take those compounds forward in terms of the medicinal chemistry and to hits to leads and lead optimization. But it's taken a while for it to get off the ground, but it's actually incredibly exciting to be in that space, having worked in both environments, and things are really beginning to happen, and the companies are beginning to share data on compounds and series, and I think it's really accelerating in terms of what's going to come through.
I think it is an accelerator. I agree, and I think Jim and Diane both, Jim's had his hand up for a while, and then let's go to Diane. I think it's a model we would like to build on and learn from as well.
Right, and to follow up on Paul's comments, I think also in the field of neglected tropical diseases, we're seeing some fresh air. I think it's worth mentioning what WIPO and BioVentures for Global Health have been doing in terms of brokering marriages between pharma and centers that do phenotypic screens. And that's been incredibly useful, because we wouldn't want to do a screen on a library that hasn't been vetted, as I think Bob Jacobs made the point with DMPK and some safety studies. And really the libraries that come from pharma are libraries which generally have had a fair amount of entry-level DMPK and safety studies, because they're usually part of some other program. So I think we're beginning to see an opening there in NTDs as well as with TB.
Please, Diane, in the front here. Just in the front here, I think, first. Sorry, I'm missing all the hands, but David after that, yeah.
Great. Thanks, Ron. Diane Worth from Harvard in the Broad. I was going to make the same comment on malaria. I mean, it didn't happen for malaria in the same kind of organized way at some level, but the same thing has happened, that, sort of essentially a phenotypic live dead screen has gone through a vast number of compounds, and that set now is very interesting for the entire scientific community. And I would say one of the things that has come out of that is, a lot of compounds are hitting the same new targets. So in fact, this assay probably in malaria, picks a certain set of targets. A certain set of targets are going to be rate limiting. But I think, again, this is an example of, bringing, compounds from diverse pharmaceutical companies for the most part, some academic compound sets, and then being able to use those to look at the fundamental mechanisms has really caused the field to leap forward. I would say that, getting past doing very small scale screens on limited libraries was the big breakthrough. And once that happened, I think the number of compounds and the number of targets has really gone up. And I think that's probably the breakthrough you need. And that's the reason for the TB Accelerator. And I think this field kind of needs the same thing. And you're starting to see it, but I do think just getting a lot of molecules that are active in killing the bugs for the acute phase kind of application is going to be a key step. And whatever we can do to lower that barrier. And somehow the companies have gotten over this issue of leaking information based on the phenotypic screen. And some of it is screening molecules that are active in their other programs. And some of it, I think, is just realizing that this is that there's so much convergence that holding the information no longer has much value.
Yeah.
Can
I think David and then Thierry, yeah?
Yeah, I'm come from a pharmaceutical background and gone through many mergers and many different CEOs. You realize that the focus within a company keeps changing. So the disease areas that the company's looking at keep changing. So then there are legacy libraries that are left behind that don't have the same intellectual property hold that they did at the time. Indeed, this is really how, through the DNDi, we found fexinidazole. It's going back through those legacy libraries that the company's no longer had an interest in. So I think organisations like DNDi need to be very acute, keep a real watching brief on what's happening within particular pharmaceutical companies, then to make an approach when the time is right.
Yeah, good point. Very good point. Thierry?
I think it will be a mix of answer for not answer but comment on your comments. I think about the TB Accelerator is really great idea, and especially what for me experience where I compared the Bio-AstraZeneca collection, there was some similarity. You see this. We say most of the two collections are different, but some parts are similar. So it's really important in terms of in project work. So because sometimes if you have a singleton, you say, I will not work. A lot of chemists, some will say singleton can be a great drug. It's okay. But most of the chemists still think that I don't want to start with a single compound. I want to see some SAR, some series, and then I can have a better idea to start what could be the next molecule. When this overlap between these compounds were similar in between Bio-Astra can be maybe we can enrich each other. So a singleton in Bayer is no more a singleton because there are two compounds in Astra very close. So they were maybe aiming for a purchase somewhere or maybe a library. Some moment during the history, two chemists had same scaffold, they put different things on it, but they comes. So I think for this kind of multi-pharma, this system, I think there is a great value of this because we will also rescue a certain part of our collection that is usually dead from the beginning, even if they eat. That's the one comment about this.
Please, Manu.
Can I follow up on that? So actually through DNDi, we're screening lots of pharma libraries in our Leishmania assay. And we're doing that blinded, and so we can't make this correlation. But just wondering from DNDi's point of view, will you have access to all the data in the structures to see if there is SAR across libraries from companies to exploit that effort fully?
I'd love to say yes, but the reality is we tend to see the structures of a few hits. We don't see all the inactives and the thousands of compounds that didn't show up. So it's a really interesting question to know, have we tested these compounds before? How do they fit? But at the moment, it's not something we can do. It's the sort of thing the TB Drug Accelerator might hope to do, but it's still a very ambitious question.
Bob Jacobs again from Anacor. I think the discussion here has moved on to some things about screening and screening for selective poisons and screening across various pharma libraries. One of the things that I would, from a couple of years of experience now, that would be very helpful, I think, to the community is as screens are done from some of the pharma libraries and from other sources, and to Diane's point of screening large numbers of compounds, it would be very helpful if the pharma, without disclosing proprietary structural information, could help assist when hit series are identified to say, this is a series that we've seen as a frequent hitter before. We've ruled this series out from projects internal to our pharma company because it's a membrane disruptor, or we know that this series of compounds is disrupting oxidative phosphorylation mechanisms, and selectivity may be an issue down the road. Again, without disclosing proprietary information, but helping to weed out those uglies that, from having been in big pharma, we all had them, we all have them. In AstraZeneca, I know we had all the Hibiclens and topical antibacterials that existed within the library. Some of those have been filtered out, but I think that would be really useful as some of these open innovation approaches take part.
One more, and then I'd like to finish on another topic, if I could. Please, Jean-Robert, yeah.
Yes, so I fully agree with Bob's comment, and this is actually the point I wanted to make back to Manu's question and request to get more information. I really think that if we could centralize, gather additional information from various pharmaceutical companies in a non-property fashion, and get a better understanding on how assays, how they translate basically, and what's the chemical space we're working with in terms of our actives. I think this will be really, again, building knowledge on data and not only access data here and there that sometimes it's really hard to understand as a whole. So that was just the point I wanted to make.
All right. I'd like to finish on another collaborative point just before, and it's probably really for people perhaps to discuss over a glass of wine in a couple of minutes. I started by talking about collaboration in the private domain. I'm also very interested, but still a little stumped in terms of some of the collaborative approaches in the public domain, and particularly I think open innovation, open source drug discovery, and I'm starting to get my head around some of it. I see open innovation as very much the same as pre-competitive research in the pharma industry, target ID, validation, and so forth. Open source is really looking at a way, pardon me, looking at a way of going that next step and getting into the expensive chemistry phase. And I think there are a couple of visionary people out there who have proposed different models. Matt Todd, I think, in Synaptic Leap, and Samir Brahmachari from CSIR in India with open source drug discovery. And they're really quite different models. One is everyone is an equal in a collaborative effort, and the community will self-select the best. The other is very much crowdsourcing, in as much as there is a head to the organization, and they dictate, or not dictate, but specify what they would like done. I'm still trying to come to terms as to whether they would work, and I think, as I say, I'll leave this for debate unless someone has something they would really like to raise now. And what I'm starting to do is educate myself on what I think are the problems or the blocks, so that we can address those and see how we can get around. And for me, the biggest one, the elephant in the room, is intellectual property. And that's not just patenting, but people wanting to keep information confidential for whatever reason, be it publication or whatever. The other, in many cases, is incentive, why people would want to belong to a big collaborative exercise. There has to be some sort of incentive. And third, for me, I call it research discipline, but not in the context of, it's really making sure the quality of the research is such that it can be an effective drug discovery program, in the sense of we see that different systems, different biological systems give quite different results, so you really need very good validation or very good centralized biology. But I'll perhaps for a couple of minutes if people have comments, but then we can perhaps discuss it over a glass of wine. Looks like you're all ready for a glass of wine. So I'll finish on one last point, and that is again to thank 6 very, very entertaining and informative speakers this afternoon. And perhaps a round of applause for the conference. And I think together with the team from DNDi, Charlotte, Martha and Gabrielle in particular, who made it possible for us to just turn up, together with the speakers, made it possible for us to just turn up on the day and be entertained. Again, I'd like to thank them for the efforts they've made. Thank you very much.